Director of the Koch Institute and the David H. Koch Professor of Biology at MIT. I've also come to think of him as our catalytic and transformative leader. As I noted yesterday, and I am mindful that not everyone who's here today was here yesterday, so I'm going to repeat my introduction of Tyler from yesterday. He is an investigator of the Howard Hughes Medical Institute, and he serves as president of the American Association for Cancer Research. So we do often wonder how he gets it all done. It's amazing. In April of this year, he was elected to the National Academy of Sciences, and in October to the National Academy's Institute of Medicine. Tyler has a lifelong familiarity with the MIT campus, as he is the son of a Sloan School faculty member. He traveled up Mass Ave to get his undergraduate degree in biology at Harvard and his doctorate in biochemistry and biophysics at the University of California, San Francisco. His graduate work was with Nobel laureate Harold Varmus, who is now the president of Memorial Sloan Kettering, is that right? Tyler was a postdoctoral fellow at the Whitehead Institute, working with the pioneering cancer research Robert Weinberg, who's one of our extramural faculty members. He joined the MIT faculty in 1992 in the Department of Biology and the Center for Cancer Research. In 2001, he was appointed director of the Center for Cancer Research, and he's guided the CCR through its transformation into the Koch Institute. Tyler's lab has pioneered the use of gene-targeting technology in transgenic mice to study cancer-associated genes and to construct mouse models of many human cancers. Please welcome Tyler. He's going to tell us about his work.
Well, thank you, Sally, and welcome again to everyone. I'm very pleased to be able to speak about my research. They usually don't invite me to do that, but I will tell you about what we do in the laboratory over the next half an hour or so. I'm pleased that we've moved rooms and we have more comfortable chairs, although the temperature is such in here that you probably wouldn't be able to feel it anyway if So my laboratory is interested in cancer genetics and the ways cancers arise and progress and respond to therapy. And we use the mouse to study those processes. And I'll tell you in some detail about some recent projects going on in the lab. But before I do, let me tell you a little bit about how cancers arise. Most of the speakers have alluded to this process, but we haven't actually reviewed it for you before now. So I thought I would do that briefly. Cancer cells arise in your body through alterations to normal cells of your tissues. And these cells acquire alterations in cellular genes which confer upon the cell new properties, specifically their ability to grow and divide inappropriately. Within that expanding mass of cells, which we might call a pre-neoplastic lesion or precancerous lesion or a benign lesion, additional changes can occur, altering still more cells in new ways, giving them new properties, perhaps to grow and divide even more effectively, or to resist signals that lead to their death, or importantly, confer upon them their ability to invade into surrounding tissue. This is a dangerous phase of the cancer process because now the tumor can move from its primary site into the surrounding tissue, and within the surrounding tissue access the blood supply as well as the lymphatics, and travel therefore to other parts of the body and establish metastases, which, as I mentioned yesterday, is the deadly phase of cancer. These alterations in most patients take place within otherwise normal cells. Most of us are born with normal copies, in fact, 2 normal copies of each of our cells, and we acquire these alterations in the course of our lifetimes. This is what we call spontaneous cancers, and as Phil mentioned, in humans we now think that these cancers acquire over the life history of the tumor perhaps 10, 20, as many as 50 specific changes in particular genes, a subset of which are contributing to the cancer's behavior. But in addition, we know that cancers can arise in individuals who have a predisposition, who have inherited a defective copy of a gene which is promoting the cancer process, or a gene which is now inactive, which would have otherwise inhibited the cancer process. These are the hereditary cancer syndromes you've heard about, such as hereditary breast cancer, where the gene is called BRCA1 or 2, or hereditary colon cancer in the APC gene. And these processes we now understand are very similar to spontaneous cancers, but these individuals start off with all of their cells having one of those requisite changes, and therefore they're one step closer to a cell which is now growing out of control, leading to increased predisposition as well as earlier onset in most cases. So this is our understanding of the genetics of cancer, which is built up over the past 30 years of cancer research. And we've defined a set of genes that contribute to this process, and they fall into two broad categories. These have been mentioned by different speakers. The first class of genes are called oncogenes, and these genes promote tumor development by, in effect, stimulating the normal cell division cycle. In fact, the normal copies of these genes, which are present in your cells, are not there to promote cancer. They're there to stimulate your cells to divide when they should divide, like when you injure yourself and you need wound healing, or during embryogenesis when a tissue is forming. These genes function to stimulate the cell division process. Likewise, there's a set of genes which we've come to call tumor suppressor genes, and these effectively act like the brakes on a cell. They act when a cell is supposed to stop dividing, when the wound is finished healing, or when the tissue is fully formed. In cancer, the alterations that we find in these genes alter their function. In the case of oncogenes, the genes get stuck in an on state. They signal constitutively to the cell that it should be dividing, despite the signals that it's getting from its surroundings that it's actually not appropriate to divide. And likewise, the tumor suppressor genes are inactivated. The brakes are lost. So that when the cell should be receiving a signal to stop, it doesn't have the mediator of that signal to carry out the instructions. Oncogenes and tumor suppressor genes are the two broad classes of genes that are mutated in the development of cancers, and there are probably a few hundred genes in our genomes which can be mutated in this fashion to drive the cancer process forward. Now, I'll be describing two genes in some detail that we work on extensively in my laboratory, both of which have been mentioned by previous speakers. One is called KRAS, which falls into the oncogene class. It's actually the most important, we believe, of all of the oncogenes. It's mutated in about 30% of all cancers. Alice Shaw mentioned yesterday that it's mutated in about 20% of lung cancer, but it's also mutated in 50% of colon cancer and 95% of pancreas cancer. This is a major human cancer gene. And P53, which is a common tumor suppressor, inactivated in perhaps 50% of all cancers. So for many cancer types, these two are major players, and this is the reason why we've chosen to study them using the systems that we work on in my lab. So we understand the progression from a normal cell in your body to a cancer through the sequential acquisition of mutations to genes that control cell behavior, oncogenes and tumor suppressor genes. And much of the focus in cancer research over the first, say, 2 to 3 decades since the focused effort beginning in the early 1970s has been to characterize these genes and to understand their function. And much of that work has been done in model systems that are quite primitive. The use of cancer cells growing in a laboratory dish, for example. And those are useful because they're facile and easy to work with, but they're not really representative of the disease process itself. And that's because cancer cells, in fact, in the body, exist in cooperation with many other cells. For example, cells of the immune system, as we heard about from Darrell Irvin, are interacting with the cancer cell in both negative and positive ways. Other cells within the environment are feeding signals to the cancer cell to divide or not to divide. Cells of the blood system, the new vessels that feed the tumor, angiogenesis that Bob Langer talked about, are also critically important. And even normal cells within a tissue can control the behavior of incipient cancer cells in powerful ways that can't be modeled easily in a tissue culture dish. Moreover, cancer cells often move beyond the primary site in the process of metastasis, entering the blood or the lymphatic system. And this, too, is a complex process involving many different cell types that can't be otherwise modeled properly in quite simplified systems. And so for that reason, when I was deciding what to do for my postdoctoral training, returning from UCSF in California to the Whitehead Institute here at MIT, I decided to study cancer and take advantage of all that we learned about cancer genetics, but to study it in an intact organism. And the organism of choice for us was the laboratory mouse. And we chose the laboratory mouse largely because we can manipulate the genes of mice with great sophistication and relatively simply. We can actually today alter any gene, in fact, any specific nucleotide of any gene, in any way, in any cell, at any time, using more and more advanced technology. That was not true when I started my postdoc in 1988, but over time, my lab and many other labs have contributed to new technologies that have allowed us to do this quite powerfully. So we've invested a lot in developing more sophisticated models, in vivo models of cancer to understand the disease process and how to control it. But actually, as a mouse modeler, I've taken some flack. And this came to a head about 5 years ago when Clifton Leaf wrote a story that was featured on the cover of Fortune magazine called "Why We're Losing the War on Cancer." And he did an analysis of the cancer research enterprise that had been built up over 30 years and which had used a lot of federal funding and basically complained that it hadn't worked well enough. And he actually made some legitimate points in that article, some of which we've responded to in the formation of the Koch Institute, about encouraging collaboration and reaching across disciplines. But if you read that article, you'll also see that a major villain in the cancer research enterprise was the laboratory mouse. And here's a quote from a scientist, in fact. "If you look at the millions of mice that have been cured of cancer and compare it to humans, you realize there just has to be something wrong with those models." Now, as somebody who built all those models, I took offense to that comment. And I actually spoke in a plenary session of a national meeting on cancer about a month after this article was published. And this person who spoke after me was Clifton Leaf. And after I beat him up, I said, in fact, you were mistaken, because you'd lumped all mouse models together, and you hadn't really appreciated the fact that there are mouse models and mouse models. In fact, what Clifton Leaf and others were complaining about was this type of mouse model. This is an immunocompromised mouse. It doesn't have an immune system. The mutation that leads it not to have an immune system also leads it not to have hair. And if that mouse were in this room, it would be frozen solid. These mice are used in cancer research, and you've actually heard some examples of it, because you can put human cancer cells into these mice, for example, under their skin, and they won't be rejected by the immune system of the animal because it doesn't have one. So this is an in vivo test of cancer cell and cancer cell behavior, but it's extremely artificial because it actually doesn't recapitulate any of those important, intricate interactions between the cancer cell and the cells of the host. They're just growing under the skin. Moreover, the cancer cells have been growing in the laboratory for 10 or 20 years, so they don't really resemble the natural tumor from which they arose anyway. So this is a highly artificial system. It's useful for some experiments, proof of principle type experiments, but it's actually extremely low bar when one is assessing therapies. So in fact, many therapies have been shown to be effective in models like this and have gone on to fail in the clinic. And we believe that one of the values of these more advanced models is to give a more representative test for new therapies. And if those therapies can pass that test, perhaps they'll have a much greater chance of working in the context of human patients. Now, mice are not humans. And this slide illustrates an important point, actually a couple of important points. The genes of organisms dictate their form and function. And the genes of mice, while very, very similar to those of humans, are not identical. And so we have to bear this in mind. Some of the things that we'll learn from the studies of mice will be applicable to the human state, and others will not. And we have to be aware of that. But this slide also shows famously in this photograph that not all humans are the same. And that difference exists because of genetic differences between the individuals, polymorphic differences we call them. And these differences actually can make the study of cancer in humans complicated, because the same alteration in two different people might have different consequences. And if one is trying to understand the fundamental contribution of that change, it might not be apparent because of these polymorphic differences between two people. And mice allow us to overcome that because mice that we grow in the laboratory are effectively clones. They are genetically identical. And so when we instigate a tumor in the background of one of these mice, the tumor is altered and changing in ways that we can study, but it's not complicated by differences between the different individual animals in which we're doing this work. Moreover, with mice, we can grow up large numbers of them. We can initiate cancers in large numbers of animals. We're not limited by sample number or access to tissue at different times. We can look early in the disease process, which is hard to do in humans. We can look late in the disease process. We can do interventions which are not practical or indeed ethical yet in patients. So the basic strategy is to use genetic engineering technologies, which I don't have time in this short presentation to describe in detail, but I'm happy to answer questions about it afterwards. As I said, we can alter the genes of mice in basically any way in which we desire, turn them on, turn them off, modify them in different ways. And we alter genes of interest like KRAS and P53 to make the animal cancer prone. And over time, the life of that animal, we will observe cancers arising in those mice, as I'll show you some examples of. And then we can study the disease in that setting. And with that as a starting point, we can do many things. We can study the details of tumorigenesis. We can look at the genomic and genetic changes that have occurred within the tumor cells as they arise. We can actually look at the contribution of the same genes to the developing embryo, where they actually often play important roles. We can look for new drugs to control the disease. We can test those drugs in more sophisticated models. And we can also use these animals to carry out better and new methods for early detection of the disease by looking for proteins or fragments of proteins in the blood, which might be an indicator of the disease process, or develop new imaging techniques, which again I'll show you momentarily. And this is one example of a mouse that we constructed about 10 years ago in the hands of David Tuvison, postdoctoral fellow, now an investigator in Cambridge, England. And David was interested in this KRAS gene, largely because of its role in pancreas cancer at the time, but we've since studied it in that and other diseases. And what David did was to construct what you might think of as a Trojan horse. He altered the gene in a way that it was activated, the oncogenic form, the flipped-on switch. But he also inserted a sequence of DNA, which we call a stop element, which blocks the expression of that gene, so it's non-functional. Rebecca Grosho, who's the daughter of one of our leadership council members sitting back here, also contributed to that work as an undergraduate working in the laboratory. Anyway, this we call a Trojan horse because it is inactive. The mouse is perfectly fine carrying this potentially oncogenic version of KRAS. However, the stop element is surrounded by short sequences which allow the stop element to be excised if an enzyme called Cre recombinase, is present in the cell. If Cre recombinase is present, it undergoes a cut and paste reaction that eliminates the stop element and turns the gene on. And we can do that with great precision in different tissues at different times. In the case of lung cancer, which we study quite a lot, we introduce that enzyme Cre by putting it in a virus, a cold virus actually, have the animal breathe the virus in. The virus then infects cells within the lung. The Cre enzyme becomes active. It snips out this little piece of DNA, and then the RAS gene turns on, the oncogenic form of it, and then we observe the consequences of oncogene activation in ways that were not possible just a few years ago. And what do we see? Well, we actually see the disease process unfold before our eyes in a matter of weeks. Initially, we observed this small outgrowth of cells within the lacy structure of the lung, this denser staining region here. This is an overgrowth of otherwise normal cells. This is this pre-neoplastic lesion that I mentioned before. We call it hyperplasia. Over time, the cells will continue to divide, probably acquiring other alterations, and form a solid but benign tumor. And then over time, they will progress further, or clones within here will progress further to develop into a more advanced adenocarcinoma. Now, with this system, we can also look at a stage of the disease process we couldn't have looked at in humans, and that is, where do the cells come from? Where do these cancer cells arise from within the lung? Is it any old lung cell that can give rise to a tumor, or is it specialized cells of the lung? And I won't describe this in detail, but Carla Kim, another former postdoc now at the Children's Hospital in Boston, discovered that it's actually a unique cell type, a stem cell of the lung, which is the cell of origin of this disease in mice, and very possibly in humans as well. Now, when we turn on RAS, we do observe the development of a tumor that gets fairly advanced. But this actually resembles early stage cancer in patients, stage 1, possibly early stage 2 cancers, non-invasive cancer. And this is not the type of cancer that is life-threatening. Instead, we observe only the advanced cancers as being truly dangerous. And so more recently, we modified the model so as to look at advanced cancers. And to do that, we needed to combine mutation of RAS with a mutation in that tumor suppressor gene I introduced you to before, P53. And if the tumors carry mutations in RAS and P53, now they will advance still further, become locally invasive, and actually metastasize. And this is a very, very severe, very, very aggressive disease in the animal, which will lead to the death of the animal within about 6 months after we initiate the process. Now, one of the goals of our laboratory is to understand what these various arrows mean. What are the molecular changes that are taking place as these genes get mutated, the ones we know about and probably the ones we don't know about? Because if we can understand that process in detail, then we have a better chance of correcting it, not just in the mouse, but in people too. And so we have a very active process of trying to understand the molecular basis of these transitions using very sophisticated technologies in biology as well as increasingly in engineering. As I said, this is a very, very severe disease. It actually looks very much like end-stage lung cancer in human patients. And I have proposed to the community that this is the test vehicle for new therapies, not the immunocompromised mouse I described before, but something with a truly advanced cancer that looks like the disease in humans. And in public meetings for the last several years, involving researchers from academia and industry, I've said for everybody who thinks that it's really easy to cure cancer in mice, remember that quote from before, cure this. And I've actually put money on the line, $10,000. I don't offer my own money. I offer other people's money. But $10,000 to anybody who can cure this mouse. And interestingly, I made this challenge about 3 years ago. It's known by pretty much everyone. It's the JAX challenge, and no one succeeded. It's a very hard challenge, and it reflects the difficulty of treating cancer in patients. But I think this does represent an important step in our ability to model the disease, including modeling therapies. Now, I've shown you pictures of tumors using histological sections that a pathologist might look at, but we can also follow tumors over time using sophisticated technologies that are also used in patients. This is a CT. You've heard of CT scans. This is a micro-CT built for mice. We have a set of devices like this that allow us to do sophisticated imaging as part of one of our Swanson Biotechnology Center core facilities. And with this technology, we can look at the disease develop over time in more illuminating ways. This is an animal in which we've initiated disease a few weeks prior. It's got a few small lung lesions. We image the mouse, then we put it back in the cage, and we take it out a few weeks later and image it again. And you can actually watch tumors evolve. And this is a quite useful thing to do for a couple of reasons. One, if you notice, not all the tumors are evolving equally. Some are growing rapidly, some are not. What's the difference between those? Does that help us explain why some human tumors remain indolent or dormant for years, whereas others are quite aggressive? What's the nature of that difference? Moreover, with this technology, we can follow the response of tumors to experimental challenges, whether it's genetic things that we do or small molecule interventions and so forth. And I'll show you some examples of that in a moment. Now, importantly, as I've tried to stress, we're really not interested in curing cancer in mice. We want to translate this information for the betterment of lung cancer patients and other cancer patients. And so therefore, it's important for us to do sophisticated comparative analyses to try to understand in what ways does this disease resemble the human disease and in what ways does it differ. And in fact, as we've looked using more and more methods, it's remarkable how similar it is, biochemically, the level of gene expression, genetic alterations, and so forth. And I'm going to give you an example of how we use these techniques to understand the disease and also how to detect it at earlier stages. This is some statistics. Alice Shaw actually showed you some of these statistics already. Lung cancer is a very prominent disease in the United States. 160,000 deaths in the world, more than 1 million deaths. The 5-year survival rate, as Alice told you, is low, about 15%. And sadly, this number has not changed in 50 years. We have had progress. Alice talked about some of it, but it isn't nearly enough. And one of the problems is that most cases are diagnosed when the disease is advanced, as she mentioned to you, after the disease has begun to spread and is much more difficult to treat by surgery and other means. And so this calls attention to the need for better ways to detect lung cancer at earlier and earlier stages. These are just some statistics that reinforce that point. And so for this purpose, we began to do an experiment or set of experiments to understand the genetic changes that take place in this model in the hopes that we could translate this information for early detection and other things in humans. And we use this device here. This is a gene chip. It's actually, I have one in my pocket. It's a small device with a 1 centimeter square glass slide, which has been microfabricated to contain several thousand probes that allow us to assess the activity of the genes in cells of a tissue, in this case a cancer. This is actually an old-fashioned one. It's about 7 years old, given to me by one of my students. They don't let me handle the really sophisticated ones. But it's an amazing technology, not developed here at MIT, I must say. But it's a very important way for us to assess the biological activity of a specimen, like a cancer. And we get data that looks like this. This has the sophisticated term, this type of image, as a red-blue pinkogram. These are a series of columns and rows which represent samples at the top and different genes in the rows. And the color represents the activity state of the gene, red being on, blue being off. And you can tell that there are many genes that are inappropriately turned on in the tumors compared to normal lung, as well as another set of genes that are turned off in the tumor compared to the normal lung. We can learn a lot about the biology of the disease by looking at those genes and knowing about their activities. But David Kirsch in my lab actually was interested in another aspect of this expression pattern. He wondered whether there were genes that were expressed that would help us detect the cancer at an earlier stage. Are there enzymes that are activated for which we could develop probes that would allow us to see those cancer cells at earlier and earlier times? And he focused in on a set of enzymes which are highlighted here called cathepsins. These are enzymes that chew up other proteins. They're called proteases. And he determined that in this lung cancer in model, and it's now known in lung cancer in humans too, the cathepsins are inappropriately activated compared to the normal surrounding cells. And this was of interest to us because a collaborator at the Mass General Hospital, Ralph Weislater, had developed a chemical probe that was silent in its native state, did not fluoresce, but was tethered to its scaffold by a linkage that was cleavable by these cathepsin proteases. And if exposed to the cathepsins, these probes were released and then fluoresced. So this was a protein-activated fluorescent probe that was sensitive to cathepsin activity. And David then started a collaboration with Ralph's lab to see whether we could see these tumors developing over time. Initially, we tested the model as I've just described it to you. We took a mouse without cancer or with cancer, injected the animal with the probe, hoped that it would disperse throughout the body, and then shined in the relevant wavelength of light, which is in the near infrared range and can penetrate tissue, and then captured the excited light on a series of CCD cameras in the hopes that we could see a constellation of cathepsin activity. This is a control animal. There's very little signal coming from this mouse. You do see a bit coming from the liver, but not much. This is the background. This is what you see in an animal with 3 tumors. They're glowing brightly. And in fact, these tumors are no bigger than a millimeter. So we're seeing them at a very, very early stage, at a stage when they could be easily removed by surgery. And we're seeing them because of a change that's happened naturally. We haven't engineered these cells to glow. We've just added a probe that gets activated by something that's altered naturally within them. We can also use this technique to follow tumor progression, an early tumor growing into a larger tumor in time. And Ralph is actually developing this technology in his own laboratory, as well as in a company, to do follow-on studies in the mouse and hopefully in humans to see whether we can use this same probe for early detection of lung cancer using thoracoscopy or bronchoscopy, where one puts a fiber optic probe into the animal, and using white light you can actually see this tumor on the surface of the lung, and using the fluorescent probe you can see that it glows. Now, I don't know if this specific probe will be used in the detection of lung cancer, or whether this target itself will be used, but this technique of learning about the biology of the tumor and developing an accurate, sensitive chemical probe, I'm sure will be, and will be valuable for early detection as well as disease monitoring in the future in cancer patients. Now, David is actually a radiation oncologist. He was at MGH and in my laboratory as a fellow. He's now at Duke University. And he wanted to look for the utility of this probe in another setting. In the case of, he treats a lot of patients with sarcoma, tumors of the muscle. And all of these patients receive surgery to remove the tumor, and then radiotherapy, and that's what David does, whether they need it or not. And it turns out that about a third of the patients don't need it, but they get it anyway because the surgeons don't know whether they've effectively removed all the cancer cells. And so they use radiation to try to kill everything in the area. And there's actually morbidity. There's a cost to that radiation therapy. So it would be nice if we could do a better job at surgical resection and in that way avoid radiotherapy where possible. And so David then developed a model of sarcoma in the laboratory using the exact same mouse actually with a RAS mutation and a P53 mutation that we could turn on with Cree recombinase here by injecting the virus into the muscle. And indeed, in these animals, within about several weeks, we observed tumors. And I took out the nasty pictures of these tumors to spare you. But the tumors arise, and we can monitor them on the surface of the animals. David first established that we could image, using the same chemical probe and the same type of imaging equipment, the tumors in these mice. And you can see those tumors glowing here. They also upregulate the cathepsin enzymes compared to the normal surrounding tissue. And in fact, we can follow the metastases of these tumors using that same technology. But what he was really interested in is whether he could translate this method to improved surgical control of the tumor. So he went back to the MGH and he asked his surgeon friends, can we work together to see whether we can use this method for intraoperative imaging of the tumor? So he developed with his colleagues a method using a handheld fluorescence camera that can be deployed in the operating room. He's working here with two MGH surgeons, Stan Hornacek and Sam Yoon, operating on one of David's mice. When they went to medical school, they probably didn't think they would be operating on mice. David is actually holding the anesthesia tube here. And the question is, can we see the tumor and the tumor borders more easily using this probe? First, I'll show you that, in fact, the tumor can be visualized through the skin using that imaging device that I mentioned before. If the surgeons take out the tumor, and using the handheld device, likewise, one can see the tumor glowing under the skin. If the surgeons take out the tumor and put it in a cassette and image it, they can see the tumor glowing. And then they go back to the surgical site. And in fact, they observe, despite their best efforts, they've left cancer cells behind. And these animals were then sewn up and returned to their cage, and every one of them had a relapse. If we could do better than that, if we could more sensitively detect those residual cancer cells, we would have better surgical outcomes and be able to avoid unnecessary follow-on treatments. And with funds from Kurt and Kathy Marble, with help from David Lee and many others, we're developing this technology, both in a company as well as in clinical trials, which we hope to be undertaken in the short term. So this is something that's being translated quite quickly. Let me now turn to therapy, and I want to frame this based on some comments that Alice Shaw mentioned to you before yesterday, and that is that fortunately, in the current era of molecular oncology, we can take advantage in some cases of our knowledge of the changes that have taken place in some genes, certain oncogenes, for example. The products of those genes can be inhibited by small molecule drugs like Gleevec and Iressa or Tarceva. These drugs combat the inappropriate function of the oncogene and stop the growth, and in fact induce the death of cancer cells. We then asked the question, using our model systems, what about the tumor suppressor genes, which get inactivated in the development of cancer? What would happen if you were able to turn them back on in an established tumor? Would that also have a positive therapeutic effect, a new way of doing cancer treatment? That is to say, is the sustained inactivation of a tumor suppressor gene required for the viability or maintenance of the tumor? And for this purpose, we designed, using genetic engineering, a special copy of the p53 gene that we inserted into the mouse. This gene is off. It's inactive in its normal state within the mouse. It has one of those stop elements that blocks its function. However, the stop element is surrounded by those same short sequences, and so with the presence of Cre recombinase, the gene can be turned on. We can put p53 back on the scene and ask whether it has tumor suppressive functions in an already established tumor. This just shows you a little molecular biology to convince you that the gene is regulatable in that fashion. I won't go through this actually, and instead just tell you the result. Oops. Okay, I think this is on a timer, so I'm just going to speak through it and it'll change itself. The experiment is to take one of these lung cancer-prone animals that has an activated allele of RAS, an activated copy of RAS, as well as the reversible copy of p53, allow tumors to develop, and then turn on p53 in these animals at different stages of tumor progression. And the question is, do the tumors respond? We've used the same imaging technology to follow the tumors, allow the tumors to grow to a size we can detect using the micro-CT scanner, and then flip on p53 and ask what happens. And what happens is that compared to control tumors that continue to grow and get bigger, the tumors in which p53 was reactivated don't continue to grow and get smaller. So even in an established tumor, the reactivation of p53 has clear therapeutic effect. It's a good thing. And this type of result has inspired the pharmaceutical industry to turn its attention to p53 reactivation strategies for the treatment of a variety of cancers. In this instance, the tumors actually don't go away completely. They're still there, even after several weeks. And this just shows you some histology about what's left. And it's actually a subtlety, but it's actually quite important. p53 in an established lung tumor clears the tumor of its most malignant cells, but it actually leaves the less malignant cells alone. It gets activated by the tumor progression process, but not by tumor initiation. So this is actually not going to be a curative therapy for lung cancer, but it still could be useful in a prevention setting. In other cancers, it probably will be curative. We had done some experiments previously in a model of lymphoma, a blood tumor, where again we could turn p53 off and then turn it back on again. The animals developed large tumors as shown here by imaging, and if we turn p53 back on and then come back and image a few weeks later, the tumor's clearly smaller. A month later, the tumor's gone, and the animal is actually cured. This wasn't true in 100% of mice, but it was true in many mice. So for certain cancers, p53 reactivation or the reactivation of this pathway, I think will be highly effective. And lastly, I'll mention some very recent work. This is actually a paper that was published yesterday from my laboratory, looking again at this lung cancer model and a pathway that's activated in these cancers that are mutant for RAS and mutant for p53 that we now believe, if inhibited, would have clear therapeutic effect. The pathway in question is called NF-kappa B. It's actually first characterized here at MIT in David Baltimore's lab. And I don't have time to describe the details of this, but to suffice it to say that this pathway, this set of proteins, responds to extracellular signals. These signals converge and convert this transcription factor, the set of proteins, from an inactive state to an active state, whereupon they enter the nucleus and activate genes which allow the cells to survive. So this is a way for cells that should be dying to survive, and cancer cells use these mechanisms to keep themselves alive. NF-kappa B had been speculated to be interesting in lung cancer and other cancers, but there was no convincing evidence, especially in really useful or really valid models. So we decided to look at it, and the way we did was to introduce into those developing lung cancers a gene which would block the activation of the NF-kappa B enzyme, protein. So the tumors developed by activating RAS and inactivating P53, but they also carried an inhibitor of this NF-kappa B survival pathway. And what we found was that in control animals that lacked the inhibitor, tumors developed as expected. But in tumors that carried the inhibitor, there was a clear effect. Clear blockage of tumors, clear regression of established tumors. So NF-kappa B inhibition has a dramatic effect in this setting in the mouse. Now we have to look in human cancers, which we're beginning to do, and we have to work with pharmaceutical companies to get inhibitors, small molecule inhibitors now of NF-kappa B that we can test in this model and ultimately in patients. And since the paper was published, I've gotten 4 inquiries to test new drugs. So I'm actually quite excited to do that. So in this brief presentation, I've tried to tell you a little bit about how we use the mouse, both to understand how cancers arise and progress, but also how we can use these models for a more practical purpose of understanding new therapies, perhaps new chemoprevention strategies, as well as early detection. So I'll stop there, and I just want to make a couple of points regarding how we do what we do and what makes it all possible. All of this work is done by students and postdocs, and I should have mentioned on the slide previously that an MIT undergraduate was one of the authors on that NF-kappa B study that I just mentioned. We have many undergraduates who work with us. Also, we're proud of the fact that our trainees go off to great places around the country and around the world, in academia as well as in industry. I also want to point out the very important contributions of the core facilities as part of the Swanson Biotechnology Center. All of these facilities contributed significantly to the work that I just showed you. We could not have done those studies were it not for those core facilities, as well as our funding agencies. And I'll stop there and happily answer questions. Thanks very much. Yeah.
Just a very small question. What is the difference between P21 and P53?
What a good question. Yeah, the question was, what's the difference between P21 and P53? P21 is a different protein. It is also involved in cellular responses to damage and actually cause cells to arrest in response to damage, not go through the cell cycle. And it's actually controlled. Its activity is controlled by P53. So when cells get damaged, they turn on P53, and P53 then acts to turn on or activate P21 and other things. So when we think about pathways in biology, P53 is upstream, P21 is downstream. Yeah?
Tyler, you and a number of other speakers have referred to the fact that combining engineering and biology and goodness many good things. But you also all refer to the fact that at some stage these things migrate into companies. What is that transformation accomplish?
The question is, what is the role of companies in the translation process from discoveries to impact in patients? And I would say it's fundamentally important, and it takes 2 major forms. Sometimes our discoveries, let's say NF-kappa B inhibition, might be useful to a company that's already developing an NF-kappa B inhibitor, and maybe they will work with us to test that inhibitor, or maybe they'll license our mouse models to test their inhibitor in their own laboratories. So that's an arrangement whereby an existing company, perhaps with an existing program, is stimulated to do things they might have otherwise not done. In addition, we often create new companies to allow us to do things that we can't do in academia. And I think probably the best example that you've heard of in the last couple of days is Bob Langer's nanotechnology targeting company, where he mentioned that they'd spun out a company called Bind in order to do the large-scale manufacture, quality control, and testing that actually isn't feasible in an academic laboratory. We're not set up to do that. We don't have the staff, space, capabilities. So the companies are formed with that specific goal in mind. Terrific. Thanks very much.